And we'll get started. So, hi guys, this is the Cell Molecular Review Session, Biology 25, and we're going to cover the pathways that we did not cover on Wednesday. So these pathways are the survival pathway, the GPCR pathway, and the JAK-STAT pathway. So we're gonna get started. If this will switch to the next slide. Okay, so we discussed in class the survival pathway. So these are just general questions to think about for this topic, and we're gonna go through all of these today. So the first thing to think about is what does this pathway inhibit? And then are you able to describe how the survival signaling pathway works? Are you able to explain it to someone who's never taken the class before? Because that would show that you have full understanding of it. And then the last thing we're going to look at is how you could cause cells to die or cell apoptosis even with survival signals present. So this is sort of like what would happen if you mutated something. Okay. So what does the survival pathway do? Survival pathway promotes cell proliferation. So the end result of the pathway is the survival of the cell. Saeed, could you monitor the waiting room in the chat for me? Because I can't
Sure.
see anything, but sounds like someone's coming in. Okay. So like I said, the pathway inhibits programmed cell death. And this pathway is important to biologists because cancer research often targets this pathway because cancer cells are bad and we probably want them to stop dividing and grow. We would want to shut down the survival pathway. And this pathway is interesting because it works together with other pathways to help cells survive. So you can see the image on the right there. I have the RAS-MAPK pathway, I believe, is there. And then I also have the cell survival pathway, the survival signaling pathway, which is up in the blue next to it. So they kind of just work in unison, kind of like crosstalk, which is what we discussed the other day on Wednesday. Okay, does anyone have questions on this? No? Okay, we'll keep going. Oh. It's not flipping to the next. Oh no. Okay, so the survival signaling pathway. Normally these pathways start with RTK. So the first thing that would occur is you have your survival signal, which will bind to the RTK, and that will cause the RTK monomers to dimerize, which means they will come together, and then they will autophosphorylate each other at the phosphorylated tyrosines that are inside the tyrosine kinase domains of the RTKs. So once that happens, your RTK becomes activated. And then you have the phosphatidylinositol, which I'm just going to call PI3 kinase. So you have the PI3 kinase, which will bind to the RTK on the phosphorylated tyrosine of the active RTK, and that is due to a SH2 domain. And so then you have the activated PI3 kinase, and then an activated PI3 kinase is able to activate inositol phospholipid. And inositol phospholipid is anchored to the cell membrane, so it's attached to the cell membrane. So that gets activated by PI3 kinase. And so initially, inositol phospholipid will start off in the diphosphorylated state, which means it has two phosphates. And the PI3 kinase will activate it. And so then inositol phospholipid goes from having two phosphates to three phosphates. So then in the triphosphate state, it's active and it becomes a binding site for protein kinase 1, which is the next part of this pathway. But I just decided to break down the pathway into steps so that you're not overwhelmed by the whole image of it. So I have So I have the small pictures that represent each section of the pathway, but the bigger scale, larger scheme picture of the entire pathway is available a few slides down. And it's important to know that inositol phospholipid is a phospholipid. I don't believe it's a protein. And there's 3 different positions on the inositol phospholipid where it can be phosphorylated at. So does anybody have any questions about this portion of the survival signaling pathway so far? I'm going to take that silence as a no, and I'm going to keep going. Okay, so then once you have your phosphorylated inositol phospholipid, this is able to bind to the AKT. So the AKT will bind to the phosphorylated inositol phospholipid because AKT has a pH domain. But the phosphorylated inositol phospholipid can also bind to protein kinase protein kinase 1, which is depicted in brown on the image. So the next part of this is the protein kinase 1 and 2 are used to phosphorylate AKT to make it active. And this is important because AKT needs to be phosphorylated by 2 other kinases to become activated. So once the phosphorylated inositol phospholipid sits in AKT, which is the green protein in that diagram, then you have to have AKT being phosphorylated by protein kinase 1. And this will cause a conformational shift in AKT, which exposes the amino acids on its surface in its binding pocket so that the protein kinase 2 is able to phosphorylate it in the second spot. So that's why you see that protein kinase 1 and protein kinase 2 phosphorylate AKT subsequently, sorry. And then the PK1 has to go first with the phosphorylating before PK2 can phosphorylate it. And like I said, protein kinase 1 is able to bind to phosphorylated inositol, PIP3, because it has a pH domain. So just picture that the cell has multiple phosphorylated inositol phospholipids, and once they become active, one can sit in the PK1, the protein kinase 1, and then another one can sit in the activated AKT. And so at this point, AKT has been activated has become activated once it gets phosphorylated by PK1 and PK2. So then at this point, your AKT is active, and it will actually dissociate from the inositol phospholipid, so which is in the pH domain at the top of the green protein, that it will dissociate itself from there and everything else, and then it will enter the cytosol. And then from there, AKT will bind to BAD, but I will get to that on the next slide. And also, I just thought I would include this because it was something that I remember she asked a question on. Sometimes she might expose you guys to P10, but just know that P10 is a phosphatase that has the ability to remove phosphates off of PIP3, which is the phosphorylated inositol phospholipid. So it can turn a triphosphorylated PIP3 into a diphosphorylated PIP3. And that's not pictured in there, but I just thought it was interesting to know. So does anyone have questions?
Yep. Yeah. So she mentioned in lecture, and I don't know if she re-specified this in lecture or in class today, but she said that after PK1 phosphorylates AKT, it will already go through that conformational shift and dissociate from PIP3 or the phospholipid inositol, and then PK2 will phosphorylate it. Is that how it's supposed to be? Because I think the information's just getting a little bit.
I mean, the way I was taught this, and I can ask Saeed as well, but I think the way I was taught this a year ago was that the protein kinase 1 has to phosphorylate AKT first, then that causes the conformational shift in AKT, and that causes AKT's shape to change in a way so that the amino acids within the binding pocket where the protein kinase 2 will bind, that area is like exposed, so it makes it so that by protein kinase 1 binding, it makes it easier, or it makes protein kinase 2 able to bind because it exposes the PK2 binding site on the AKT. Once those protein kinases bind to AKT, then at that point, the AKT dissociates from the inositol phospholipid, and then it goes downward. It goes onwards and dissociates from inositol phospholipid and everything, and it travels to the cytosol. That's the way
Yeah.
I was taught that.
Yeah, that's exactly how I understand it too.
Yeah.
Okay. So then I think it might have just been some misconception when people were asking about it, but it stays bound to PIP3 until both PK1 and PK2 finish phosphorylating.
Yeah, because
Yeah.
PIP3 is anchored into the membrane there. So PIP3 doesn't really move, but AKT dissociates from everything once it becomes fully active. But remember, you don't get an active AKT until you have PK1 binds first on the left, then PK2 binds second on the right. So once both of those kinases have done their thing, then AKT is active and it floats down to the cytosol to the rest of the pathway, and then it will bind to BAD and do the bottom half of the pathway, which I will get to on the next slide. But that's how you get an active AKT. You won't have the active AKT until both the PK1 and PK2 have done what they need to do. Does that make sense for that?
Yeah, that makes sense.
Okay, cool. Are there any more questions on that? I'd be happy to answer any more questions.
Actually, do you have one question?
Mm-hmm.
Is the bad protein and BCL2 the same thing?
No, they're two
No.
separate things, but I'm going to get to it because I didn't want to put the whole diagram in there, and I felt like it'd be a bit overwhelming, but I'm taking it step by step here. So this is the activation of AKT step, but that's coming up on the next slide.
Okay.
Yeah, so I'll definitely answer your question. You'll see. Okay, so no one else has questions on this slide specifically. I can move to the next one. I just can't tell because I can't see the chat, but if someone could give me a heads up.
You're good, Michelle.
Okay, cool. Okay, so once you have your active AKT, you can see that the PK1 and PK2 have phosphorylated it and made it active because you see the two yellow phosphates on it. So then your active AKT dissociates from the inositol phospholipid, like we said, enters the cytosol. Then inside the cytosol, AKT is going to bind to BAD. And BAD is a protein in the cytosol. So now this is where it gets a little bit tricky because you guys at this point have learned that when you phosphorylate things, you activate them. But this is different in the case of BAD protein. So active AKT will phosphorylate BAD, which causes BAD to become inactive. And so once this occurs, you have inactivated BAD, which is phosphorylated, which you can see in the diagram here. But normally, BAD is active in the cytosol, and it is binding to a different protein called BCL2. And BCL2 is a apoptosis inhibitor protein. An apoptosis inhibitory protein means that this protein is a protein that prevents cell death, so stops cell death from happening. So normally, you have BAD, which is bound to this protein. So as long as BAD is bound to BCL2, then BCL2 is unable to do its job. So BCL2 needs to be free. So this is why an active AKT will phosphorylate BAD, which basically hinders BAD from being able to hang on to BCL12. So this will free BCL12, and BCL12 will swim on its way to be free, and it will actively prevent programmed cell death. And it does so by binding to the mitochondria, and there's some other things that BCL2 does, but you don't need to know that. You just need to know that BCL2 breaks free from BAD, and it's able to prevent programmed cell death. But normally it's inhibited by BAD. So that's why AKT phosphorylates BAD, makes it inactive, so that BCL2 can do its job. So then, because BCL2 is free to actively inhibit cell death, the cell is saved from cell death, so the cell survives. I'm also going to explain BAD and BCL2 a little bit more in the next few slides, because I know that can be a little bit tricky to understand. So does anybody have questions so far on this slide? No? Okay. I'll just keep going then. Okay, so I'm just going to explain a bit how BAD and BCL2 work. And so we have apoptosis, which is cell death. And death is the cell's default setting. So that's why when you hear us say programmed cell death, that's what the cell is like programmed to do. And so BAD is a protein that initiates apoptosis, and then BCL12 is a protein that inhibits apoptosis, meaning that it will save the cell. So there are two things that can happen in a cell. So you could have an active BAD, which is always bound to BCL2. And so because BAD is bound to BCL2, BCL2 is not free to go and save the day and promote cell survival. So as long as BCL2 is bound to BAD, it's in its inactive state. And so this can cause mitochondrial holes, which eventually leads to cell apoptosis, which is a bad thing, hence the sad face. And then in the cell survival pathway, AKT will phosphorylate BAD, which renders BAD inactive. So BAD becomes inactive, and it's unable to bind to BCL2 when it's phosphorylated and inactive. So then BCL2 is active at this point, and it's able to break free from BAD and inhibit apoptosis, meaning that your cell ends up surviving and living, which is what we want. So it's a bit confusing, but basically BAD inhibits the inhibitor, inhibits the apoptosis inhibitor. But yeah, I have more slides on BAD and BCL2 and diagrams, which I think will help. So does anybody have questions on this so far? No? Okay, I'm gonna keep going. Yeah, you can switch to the next slide. Thank you. So this is just a diagram of what I was saying. And so you have initially BAD is bound to BCL2, and the inhibitory protein is inhibited. So the thing that inhibits cell death is inhibited, so you get cell death. That makes sense. And then scenario 2 is what you have when your survival pathway is working properly. AKT comes along, it phosphorylates BAD, which makes it inactive. And then because BAD is inactive, it's unable to bind to BCL2, because you can see in the diagram, inactivated BAD binds to something else. So when BAD is inactive, when its phosphorylated state, can't bind to BCL2, so then BCL2 is able to dissociate from BAD successfully, and BCL2 becomes active, and it can save the cell from dying by preventing programmed cell death. So I hope after I said it a few times that it makes a bit more sense that BAD and BCL2 are different proteins and how exactly BCL2 works to save the cell. Does anybody have questions on this? No? Okay. I'm gonna keep going. And so I included the diagrams of the entire pathway to give you guys the big picture idea. And I highly recommend that when you're studying for exams and stuff, that you come back to slides like this that don't have any words on them, and you just walk through the pathway with yourself, or talk to someone who isn't in the class or a pet or something, and just like explain the steps of the pathway, just so that like you know that you understand what's happening in each step and what's activating what and what's being deactivated and what types of domains are in there, because it gives you a good sense of what happens in a normal pathway, so that when Professor Francis asks you questions like, what would happen if I um, deactivated AKT, or if what would happen if I removed the signal, you could answer sorts of questions like that when things kind of go abnormally in the pathway, because she likes to ask questions like those in exams and problem sets.
I have a quick
Yeah,
question.
this is just a summary of the pathway. Yep, I'm sorry. You can ask your question.
They're good. So the activated receptor tyrosine kinase, right? If that was switched to a different amino acid, and I think in class today they mentioned phenylalanine, would that just inhibit all the downstream processes because these RTKs will only work unless there's a tyrosine kinase domain?
I'm not really sure because she's never thrown us something like that. She's
I
never really mutated the tyrosine kinase. She's usually mutated the other proteins within the pathway. She's never asked us.
I think it would, because
Like it
the
would.
RTK can only work with the tyrosine
Yeah,
if it
because of
switches.
the dimerization and the autophosphorylation of your RTK only works because the tyrosines within
Mhm.
the tyrosine kinase domains within that RTK receptor, those are the things that are cross-phosphorylating each other. So when you have those phosphorylated tyrosines, that's what sort of like starts the whole show or the whole pathway. So I'd imagine that without those, you would predict that nothing really would happen. That's what I think, but I don't think she's never really asked me that. I would remember if she had asked me something like that for an exam. I don't know, Saeed,
I
did she?
figure like if there was a difference in like tyrosine to phenylalanine or something in that domain, would the RTK still be able to dimerize? It's just like the tyrosine kinase wouldn't become phosphorylated, so it wouldn't allow the activated PI3 kinase to bind. Like
Well,
would the
I
signal still
believe
allow them to
that
dimerize or no?
because signal causes your dimerization to happen, correct?
Yeah.
Mhm.
So then
So
I think your signal would bind, the RTK monomers would dimerize, but then maybe there'd be some issue with cross phosphorylation. So your RTK never becomes active. And so I'd say you'd be correct that the downstream events, all that stuff after you get the phosphorylated tyrosine on the RTK, all the rest of that stuff wouldn't happen. I think
Okay.
you're correct in assuming that.
Okay. I just wanted to double check because something like that was mentioned today in class.
Okay.
I
I
have
can follow up with her on that, but I don't remember being asked that on the exam where like she switched it from tyrosine to phenylalanine, because most of these things operate with tyrosine kinase, RTKs, most of the pathways. And I heard someone had another question.
Yeah, I just want to know, is it important to know that the domain types, like if it's a SH2 domain or something?
Yes, so domain types are important, I believe, because she has asked things like that.
Mhm.
But for the most part, in this survival pathway, so you notice that the other pathways we have SH2 domains sometimes, right? But for this pathway, we have pH domains and SH2 domains. So an example of an SH2 domain is right over here. And an SH2 domain is when you have like a phosphorylated tyrosine that creates a binding site for another protein. So here you have the SH2 domain of the RTK, the active RTK, creates a binding site for activated PI3 kinase. And then here we have pH domains. So these are different because these are binding sites specifically for phospholipid inositol with the 3-phosphate. So you can see how it's kind of shaped like with the 3-phosphates, because those are specific binding sites for PIP3. So AKT and protein, excuse me, I think that's protein dependent kinase 1 or just protein kinase 1. So protein kinase 1 here and AKT, they have pH domains. Yeah, so it's important to know the domains for the pathways, I believe. And I do believe we covered some of those on Wednesday. So each pathway will have like a different set of domains, I believe, but most of them, whenever you see phosphorylated tyrosine, just think SH2. And there's plenty of examples of SH2, I think, in the RAS MAPK pathway, as well as some of the other ones. But I'll make sure to highlight some of the domains as we go through the rest of them.
Okay, thank you.
Any other questions? No? I'll keep going. She likes to ask about domains though, so I just think it's important to know where these certain domains are. Cool. So this is just a summary of everything I just said. Some people are visual learners, or some people, they like to learn things in a list. So I just have the list written out, but it's pretty much everything I just said. I don't feel the need to repeat it, but I just thought I'd have it laid out for you in that fashion. And then the way I had it laid out before with the diagrams of like the cross section of it, where there's like each of the little diagrams represent the different steps of the pathway, and then the big picture that has the entire pathway. So I just thought I would lay it out this way for people who like to have a list of everything that happens all at once. Okay, does anyone have any questions on this? Yeah, so this is just a summary. So I'm going to move to the next slide if nobody has questions. Cool. And I don't know why I included it again, but this is just the entire pathway. I shouldn't be in there twice, but okay. Going to move on.
Okay, so now we're going to go over the JAK-STAT pathway and then afterwards the GCPR pathway. So, so to start off, what you want to be thinking about in your mind when we go over this is, one, is JAK-STAT considered to be an RTK? And also, how does the pathway, like in general, work? How does each step lead to the next one? And if you were to knock out a step, how would that change either the upstream or the downstream effects? Okay. So before we start, does anyone have any questions or no? All right. Okay. So a quick recap. RTKs are monomers with an extracellular receptor binding domain and an intracellular tyrosine kinase domain. So it's important to realize that the binding and the kinase domains, or the binding portions and the tyrosine kinase portions, are domains of the same receptor. And also a kinase is any enzyme that catalyzes phosphorylation. And in this case, we're going to deal with phosphorylation proteins. So again, RTKs work by causing a signaling molecule will cause a dimerization of two RTK monomers, whose then tyrosine kinase domains will interact to stimulate kinase activity. And this causes autophosphorylation, so they'll phosphate themselves, which will then be used as a binding site for other signaling proteins and carry on the pathway. Okay, so with that, if we go to the JAK-STATs pathway, it's involved in many processes like hematopoiesis, immune response, mammary gland, lactation, and fat tissue generation. The example we look at is going to be for lactation, so prolactin. And some important things to remember is that JAK, the receptor itself, is not a RTK, because if you go back to the other slide, it has to be a receptor with the kinase domain. But in this case, the difference is that JAK, it's still considered a receptor, but it doesn't have a kinase domain. It has subunits attached to it. So if you look at the diagram here, you'll see the receptors themselves, and they don't have a domain. They have subunits, which are like separate from the receptor, but they're just like closely connected with the larger complex. So in that case, you can consider it an RTK, even though it does pretty much have like a similar like analysis function. And again, some quick facts about JAK. Just stands for another kinase. An easy way to remember the function is if you think of Janus kinase, if you think about that, the two-faced god, you need to have two of them to come together and dimerize. And yeah, and again, they're not kind of RTK. So if we go through it, yeah. So basically, you're gonna have two JAKs, JAK 1 and 2. Each of them is gonna be bound to its own prolactin receptor. So basically, the ligand's gonna come in, it's gonna go into the receptor pocket, which is gonna cause it to dimerize. So when the two receptors dimerize, it's not shown in like obviously like a two-dimensional like diagram, but when that happens, the two receptors will dimerize, come together, and that'll allow JAKs 1 and 2 to like touch, which will then allow them to cross phosphorylate each other, and that will allow it to then phosphorylate the receptor itself. And now that the receptor right here is phosphorylated, this will allow STAT5 protein to come in, and because STAT5 has its SH2 domain, it'll be able to come on and attach to the receptor via the phosphorylation. And then a point that I always forget is that when this happens, then again, the JAKs will then phosphorylate the stats right here. So now you have the stats, each of them phosphorylated twice. And now when that happens, then the two STAT5s will then dissociate from the receptor, and then they'll dimerize with each other. And then basically when that happens, then the complex is allowed to enter the nucleus with some other transcription regulators, and then it can start transcription of milk protein genes. So yeah, that's pretty much it. It's pretty straightforward. So yeah, do we have any questions about the JAK-STAT pathway?
I just wanted to cut in real quick
Yeah,
and talk about the domains we see here. So there is an SH2 domain. Saeed, if you want to do the laser pointer thing, there's SH2 domain on the receptor that allows the STAT protein to bind there. And then when those two STAT proteins become active and they dissociate from the receptor and form this dimer, they're connected together by an SH2 domain between them. So those SH2 domains are interacting to hold it together.
right.
And that's where the domains are.
So if you don't have any questions, I'll keep going to the next slide. And yeah, just another summary. These are all the steps. And again, it's important to know that while it does have the same functions as an RTK, JAK itself is not considered, because the main difference is that while an RTKs have domains that allow the kinase activity to happen, JAK is considered a different subunit. It's just attached or in close relation to the receptor. Okay, so if we don't have any questions, we can go on to, oh yeah, and then, yeah, so basically, if you can explain any part in this pathway, and if you can like tell, oh, so if like you knock out the SH2 domain here, for example, like what's that going to affect, then you should be all set for the exam. Going
Just
on,
pretend, just pretend like you're telling somebody who has no idea or has never been in this class what's going on. If you can do that, then you're solid.
yeah. And also remember, there's a lot of like small steps here, like where here it'll cross phosphorylate, then it'll phosphorylate the receptor, stats comes in, and then it also gets phosphorylated. So there's a lot of little steps like that that you got to memorize. But as long as you're good with that, then you should be all set. And now we have
one question I have for the other one. How does phosphorylation happen?
how does osmolar? I'm not sure exactly, but if I had to guess, I think because it has the kinase domain, then it'll be able to catalyze or yeah, act as an enzyme and then phosphorylate itself. I think, but I'm not sure.
I don't think for this purpose of this class, you don't need to know the specifics of how autophosphorylation happens. You just need to know where it happens and what does it. So for the JAK stat, know that it's the JAK proteins in the light blue that are able to cross-phosphorylate each other or autophosphorylate each other. And when that happens, that's when your receptor becomes active, and an active receptor allows the STAT5 proteins to bind there at the SH2 domain. So yeah, you don't have to know exactly the process of autophosphorylation because I don't even know how that works. So I don't think you have to know the specific on that.
Okay, thank you.
Sure.
Now, G protein couple, or G protein coupled pathway. So basically, when we're gonna go through this, like again, you just gotta keep in mind, can you explain all the steps to yourself, to someone who has never taken the class? And a good way to know if you understand the material, if you can explain it in a very simplistic way. Like, well, you still know all the details, but do you really have an understanding to do that? And another thing to remember in this is that you're gonna want to be looking for um, analogous function. So in this pathways, there's no gap and there's no gap, but there's still going to be pieces in the pathway that are going to be performing basically the same function. So you're going to want to know. I know for when we took the exam, she asked a lot of questions about this part right there. So onto the reaction, we have a diagram there, and I think we have another one. Oh, we have the steps. Okay, so what we can do is, or I'll just explain it here. So yeah, basically, with this pathway, you're gonna have a 7 transmembrane protein, and basically, that's gonna form like a sort of a log, and that's gonna be attached to a heterotrimer. So basically, 3 subunits that are different from each other. You're gonna have your G alpha, your G beta, and your G gamma. And it's important to know that you can kind of see it here. The G gamma is going to be attached to the membrane as well as the G alpha. And that's important to know because when a signal comes into the to the 7 transmembrane, it's going to induce a conformational shift in both the membrane itself and the G alpha. And when that happens, it's going to cause 2 things to happen. So one, it's going to create a conformational change in the G alpha, and that's basically going to want it because it's so the G alpha in this pathway is going to be the GTPase. It has the enzymatic activity, so it's going to be bound in the inactive state to GDP. But when a signal ligand comes in, it's going to cause a change in structure. And that'll cause the GDP to dissociate, and when that happens, just because there's a lot more GTPs in the cell, it's going to make that more, it's going to increase the affinity for GTP to come in. And when that happens, uh, G alpha is going to dissociate from the other 2, you can see here, because again, it's connected to the membrane. So in a lot of pathways, after dissociation, this complex is going to have its own effector, and this complex is going to have its own. But for the purpose of this class, I think we only focus on this aspect. So yeah, so when that happens, GT or G alpha is going to have GTP, and it's going to attract an effector, usually cyclic AMP, like as you know, is like a popular second messenger. Yeah, and that's going to happen. So this will basically cause the activity to happen of the activation of the GTPase. And I think this is considered a negative feedback loop. So the downstream effects, it's going to cause RGS to come in. And then when that comes in, it's going to bind to the G alpha, and it's going to increase the G alpha is already, so G alpha is going to be a GTPase anyway. It's going to be hydrolyzing the GTP on its own, but at a much slower rate. But when RGS comes in, it's going to speed that process up, and it's going to allow the GTP to hydrolyze much faster into GDP, which is basically going to bring you back to where you started. It's going to deactivate the G alpha, cause it to come join with the other 2 again, and then basically that just starts off the whole pathway again. If we go to the next slide, that's just, oh, I can't, oh, there we go. Yeah, basically that's just a summary. And the important thing I hope you picked up is that the GPCR is the Jeff analog and the RGS is the gap analog. So yeah, is there any questions about this pathway? No? All right. Here again, we have just another like example. This is just the same thing that we went through. Um, a ligand's going to come in, a signal, and that's going to allow a conformational change in the alpha subunit, which is going to take out the GDP, and it's going to bring in a GTP, which is going to basically allow it to disassociate from the other two, which can cause an effector to come in, such as like cyclic AMP. And then when the cell's done with that, then RGS is going to be recruited to just increase G alpha's already GTPase activity. So it's going to hydrolyze the GTP into GDP, and that's going to bring you back to where you started. And that's just another example or another diagram of the whole thing. So some common misconceptions, the gap and Jeff function like we're talking about. I know that she asked, I remember these questions a lot specifically. She will ask about enzyme activity and what are and aren't enzymes. It's important to know that gap and Jeff, they're not enzymes because they don't have any catalytic properties. They just assist the actual GTPA or GTPase, but they're not enzymes themselves. And Jeff, promotes the GDP to dissociate and allows the GTP to diffuse in, or at least increases infinity. And then the gap analog is basically just going to accelerate the proteins already in a GTPase enzymatic activity. So yeah, that's pretty much that for any common misconceptions.
So, would you say that the enzymatic activity in this pathway is done by G alpha? Like the
Yeah.
only thing in, okay, so G alpha would be the protein that has this enzymatic activity. But
Yeah,
okay.
yeah, exactly. That's going to be your protein or enzyme.
Would the beta gamma complex have it as well, or is it only the G alpha?
That's a good question.
On G alpha for this class. When the
Yeah.
G alpha dissociates and goes on to do signaling, then pops back in and attaches itself. We don't look at the other two really.
Yeah,
Okay.
yeah. For this, we just want to focus on the G alpha.
Okay, so it's just G alpha that has the enzymatic activity.
Exactly.
Okay.
Yeah.
Sorry.
No, that's fine.
I just wanted to say that, yeah, sometimes when people write down their answers, at least for like open response and stuff, people will say that GAP hydrolyzes GTP, but that's not really true. An example of this is, if you guys remember way back when we did the nuclear import and export, you saw that GAP was in the cytosol, and so GAP was attaching to binding to RAN, and it was helping to speed up or accelerate its RAS's own hydrolyzing activity RAN's own intrinsic GTPase activity, because RAN by itself is an enzyme and that has the ability to hydrolyze GTP and GDP. So yeah, that's just something I thought I would put in there.
Yep. So yeah, that's pretty much it. Let's see what we have next. Yeah. So that's pretty much it, guys. Yeah, all we can really say is you just gotta practice these pathways. They're pretty straightforward, It's I don't want to say it's a lot of memorization because the way she sets up the exams, as you guys know, is a lot of like, if I change this aspect, if this has a gain of function, or if this part has a loss of function, how is that gonna affect the downstream? So as long as you know all the like steps and all how they like lead to the next step, you should be perfectly fine. But yeah, I hope this like cleared up any questions or solidified your understandings.
I'm sorry for asking so many questions, but I'm having a lot of troubles understanding this. And like I was doing the homework and I didn't understand the last part, and I thought that this would help me understand